Head principal and lecturer. Immunity pathogens produce antigen and the organism form the and fight against immunity and studies called immunology. First is innate non-specific immunity, physical barrier by skin and mucous membrane. Physiological barrier number 2, number 3 lysosome form lysozyme and gastric juice form gastric juice destroy pathogen. Cellular barrier only polymorphic fungus form GAMS and neutrophil are present and monocytes form natural killers. Kills to phagocyte cells. When these make pseudopodia weak and they call the update pathogens and neutrophil form monocytes, macrophage form skin monocytes and skin form Kupffer cells and Fiji cells and protect the organs. Now we see in this diagram we are looking the phagocyte cells. Phagocyte cells forms phagosome, then phagosome form pseudopodium and this is called cellular barrier. Now phagosomes monocytes Kupffer cells. Now we proceeds to the
पढ़ रहे आदमी दूर पढ़ रहे
barrier. In cytokine barrier, the viral nucleic acid attack on host and it attacks on DNA. Then some cells are formed, new virus are formed. This is interkinin. Then mRNA is formed and the interferon molecules are formed. These molecules again attack on the cell and they form नीचे कर थोड़ा सा
कॉमन में चल तो बोलते रहे हो तुम
समझ में आना में दिखाई नहीं दे रहा ना
लाइट बाड़ू ना
हाँ लाइट बाड़ most cell makes interferon and the most cell to produced against virus by interferon. From cell 1 to cell 2 they enter in the interferon cells to form the or and they attack on the chromosomes. Then they cells from the new viruses they attack from the cell 1 and antiviral proteins block viral reproduction. Now that B lymphocytes and T lymphocytes formation. In B lymphocytes and T lymphocytes, bone forms lymphocyte cells and then they form B cell and they storage in lymph node in lymphoid tissue. And thymus gland T cells form T cell and T cell then form cell mediated immunity. It is very important. Now we proceeds to the humoral immune process and cell mediated immune process. It is very important. First of all, we see that antigen test exposure, then antigen presenting cell is formed, then helper cell is formed. Helper cell on the one side it forms B cell, on the other side it forms cytotoxic T cell. Then memory helper T cells, memory T cells form antigen again, second exposure. Then they form memory B cells and some B cells come from the lower side and they form the plasma cell. In again cell they form memory cytotoxic T cells and active cytotoxic T cells. Then we see again active cytotoxic T cells. We see ये पूरा हो लिया. See the structure of antibodies. Antigen form A cells, B cells, and they are protein molecules. Antibodies contain proteins. They contain 4 peptides cells. 2 cells are called light chain and 2 are called heavy chains. And they contain disulfide bonds. They contain disulfide cells. And TCA and the antigen binding site you are looking in this diagram, and constant region of light chain you are looking in this active and passive immunity. Active and passive immunity, after passive immunity after disease. It contains disulfide bonds. Okay, boy?
बोल रहे हो रहे हो
Vaccination and immunization. After active immunity, antibodies are formed. Antibodies protect disease. Vaccination protect infection. Vaccination prove the antibody difference between active and antibody. In active and passive body, we can differentiate. This is very important for examination. This is produced by vaccination or by contact. Not produced by vaccination, not by vaccination. This is produced by vaccination. It is not produced by vaccination. Not produced. Body antibody is prepared. Not produced. Not prepared. Not given to weak person, and it is given to the strong person.